Hello, my name is Paula Tourinho, and I'll present the work entitled Microplastic Fibers as a Vector of Silver Nanoparticles, Adsorption Mechanisms, and Ecotoxicology. I'm a postdoc in the group of Ecotoxicology and Life Cycle Assessment from the Department of Environmental Chemistry, led by Professor Vladimir VŠCHT. Ecotoxicology is an interdisciplinary field where we try to answer questions regarding the toxicity of compounds and also including their route of exposure, environmental distribution, mode of action, and so on. So to answer these questions, we do experiments in the laboratory where we expose animals to different concentrations of our contaminant, and we access endpoints such as survival, reproduction, and growth. We end up with a relationship between dose and effect. So here, for example, you can see in this graph, with increasing the concentration of the contaminant, we decrease the response in the organisms. And this is very useful if you want to compare exposures or contaminants, because, for example, the compound in the red line, you have the same response, but obtained at a lower concentration of the contaminant. Therefore, you can conclude that this compound has a higher toxicity in comparison to the compound represented by the black line. There are different levels of biological organization where these effects can be observed. And all starts with the exposure, which will affect first the lower levels, starting with the molecular level. So we'll have effects on protein expression, gene expression, DNA damage. It will be followed by effects on the cellular level, such as energy allocation, the functions of the organelles. Then will affect the individual level by reducing the reproduction, the growth and survival, changing their behavior. Then you have the effects on the population and community level by changing population structure, the abundance of the species, and also its diversity. And finally, you have the effects on the ecosystem level. For the past 2 years, I've been working this project called ToxMina, the combined toxicity of microplastic fibers and silver nanoparticles. So here today I will present some results we obtained in this project. Microplastic, they are generated by the breakdown of large plastic items, and they are defined as particles having less than 5 millimeters. They gain a lot of the public attention lately because they are being found in oceans, lands, and freshwater systems, so they really become a worldwide problem. The microplastic fibers are a specific type of microplastics, and they are generated when we wash synthetic textile in our homes. And then these fibers, they will go to the wastewater treatment plants and be found in both effluent and sludge. So in this way, they will reach the aquatic and the terrestrial environments. In the same way, the silver nanoparticles are being incorporated in textile due to their bactericidal properties, and they will have a similar pathway as the fibers. It raised questions regarding the interaction of these two contaminants, how the microplastic fibers will interact with the nanoparticles. Can they absorb these particles? Or how it will affect their toxicity? So in the first part of the project, we analyzed the sorption of the nanoparticles to the fibers. We use citrate-coated nanoparticles, and we expose together with our fibers, and after a certain period of time, we would filter the water in order to extract the fibers, and we analyze the silver content in the water. So as you can see, after 48 hours, approximately 35% of the silver was found to be absorbed by the microplastic fibers. Also, it took 6 hours, so the equilibrium was reached. And then we conduct a second experiment where we change the concentration of the nanoparticles in the water, and we found that the quantity of the nanoparticles adsorbed to the microplastic was non-linear with this concentration. Therefore, it indicates that a surface adsorption was the main mechanism. Therefore, to elaborate a little bit more on the mechanism of sorption, we must understand the speciation of both nanoparticles and the fibers. So starting with the fibers, these polyester fibers used in the study, they have this carbonyl group, which are very likely to produce negative species when in contact with water. Also, the nanoparticles in the water phase, they will oxidize and have a positive surface charge, and this surface charge will attract negative ions, which will form what we call a stern layer. The nanoparticles can dissolve into ions, releasing these ions to the solution, which are free to interact with the negative surface of the microplastic fibers. In the same way, something called ion exchange can occur, in which one of the ions from the stain layer will be replaced by another negative ion, and in this way, the silver nanoparticle can also interact with the negative surface of the fiber. Therefore, we propose that electrostatic interactions are the main mechanisms for the sorption. In the second phase of the project, we analyzed the ecotoxicity of this combination. And first we start with the aquatic one using the water fleet Daphnia magna. And then we expose our organism to citrate or PVP-coated nanoparticle alone or in combination with the microplastic fibers. We put 5 animals per jar, and after 48 hours we analyze their survival, and also in this case we analyze the cellular energy allocation. So here you can see the number of surviving animals with the concentration of the silver nanoparticles in the water. And for the citrate-coated nanoparticles, in black you have the nanoparticle exposed alone, and in red in combination with the fibers. And in the same way, for the PVP-coated in the right graphic, you can see the black line indicated by the nanoparticle alone, and the dashed lines for the combination with the microplastic fibers. And in both cases, we observe the shift of the curve to the left, which means that the toxicity of the nanoparticles was higher in combination with the fibers. The cellular energy allocation was evaluated by the energy available in the cells, which was calculated by checking the levels of carbohydrate, lipids, and protein. And overall, there was no difference in the energy available between the control and the treated animals. However, when we look at the energy consumption, we did observe an increase in the energy consumption in animals exposed to microplastic fibers alone or in the combination of the nanoparticles and the fibers. So the Daphnias, they are filter feeders, which means that they will filter the water and retain the suspended particles. This is a normal situation where they will retain, for example, the food, in this case the green algae, and then you can see the gut completely full with the food. Here I show pictures of the Daphnias from our experiment. In the left you have the control Daphnias, you cannot observe its gut because it's completely empty. On the right you can see the Daphnia exposed to the microplastic fibers, and the gut is completely full with the microplastic fibers. Therefore, we conclude that this increase of the toxicity of the combined exposure is related to ingestion of the microplastics by the Daphnia, which are adsorbed with the silver nanoparticles. And for the terrestrial ecotoxicology, we use their form Eisenia andrei. For that, we expose the worms to PVP-coated nanoparticle in the presence and absence of the microplastic fibers. So we put 10 worms in each jar, and after 48 days, we measure the reproduction by counting the number of juveniles, and we check also the biochemical changes. So here again, you can see the graph for the effects of the nanoparticle alone, represented by the black line, and the nanoparticle in presence of the microplastic fiber, represented in red. Again, they shifted to the left, indicating a higher toxicity of the nanoparticle in combination with the microplastic fibers. When we check the biochemical profile, we could see that when we add the microplastic fibers and both the fibers and the nanoparticle together, a different profile was observed by the worms. There was some difference in the content of protein, lipids, and carbohydrate in general. So as a takeaway message is that the microplastic fibers, they can interact and they absorb silver nanoparticles. Therefore, they can really act as a carrier of the nanoparticles in the environment. And also that there was an increase in the nanoparticle toxicity in the presence of the microplastic fibers at both cellular and individual levels. When I first started the project, I got a very interesting question from the audience that was, what can we do regarding the plastic pollution? Well, specific for the case of microplastic fibers, there are a number of products now in the market that promise to decrease the number of fibers released to the environment. There are filters you can connect to your washing machine, or even there is a first washing machine who has already a specific filter to obtain these microplastic fibers. However, the efficiency of these products are still in question. So overall, for all the microplastic that are produced, what we can do is, first, we can be more responsible consumers. That means that we should decrease the amount of plastics we consume and choose for more natural materials. And second, we can also put pressure through our politics and government and lawmakers in order to make them to take action regarding this plastic pollution. I would like to thank all my VŠCHT partners and project collaborators. I invite you to check our website, and if you have any comments, suggestions, or questions, you can please write me. And thank you for your attention.